All right, Ninja Nerds, so in this video, we're gonna continue on with what we finished up with. So if you guys remember, we talked in the recent video in part 1, which if you guys haven't seen that, you guys gotta go and watch that first, okay? Then we're gonna go over in this next video, we're gonna talk a little bit more about the myofibril. So in the recent video, we took a skeletal muscle, right? We took that belly, we made a transverse cut, we looked at that transverse cut and we saw the connective tissue coverings surrounding each layer, right? So for example, the muscle belly is covered by a connective tissue called the epimysium. Then inside of that muscle belly, we have these things called fascicles. And fascicles are just bundles of muscle fibers. And then each one of these are a fascicle, right? And then around them, around that fascicle, you have a nice connective tissue covering, which we showed right here. That was called the perimysium. Then in this fascicle, there's individual muscle fibers. What we did is we pulled one of those muscle fibers out, which again, you can call muscle fiber a muscle cell. There's a connective tissue covering that muscle fiber, which is called the endomysium, which is an areolar connective tissue. And then we said that you have a lot of this filamentous endoplasmic reticulum structure, which we call the sarcoplasmic reticulum. It's a nice calcium storage factory. We said that it's striated, which means it's striped, and that's due to this actual connection of thin and thick filaments that we'll talk about, which is going to be making up the basic functional unit called the sarcomere. And then we said it's multinucleated, and we said that the muscle fibers are cylindrical. And we said each muscle fiber is surrounded by a plasma membrane, which we call the sarcolemma. Okay, then we took, and there's thousands, hundreds upon thousands of these myofibrils, which are just many, many different connections of thick filaments and thin filaments and accessory proteins that we'll talk about, and that is our myofibrils. And what we did is we zoomed in on a portion of a myofibril, and now we're gonna talk about that. All right, so first thing that we're gonna do is I'm gonna lay out some different zones and regions and stuff like that, because that's gonna help us to continue to keep narrowing down protein after protein, because literally that's all this whole basic myofibril is composed of, is proteins. So let's do that first. So the first thing I'm gonna do is, if you see this pink zigzag protein going all the way up, this protein is actually going to be called the Z disc. Okay, that's the Z disc. And we'll talk about what protein it's made up of in just a second. On this one over here, this is another zigzagged protein, and we're going to call this one a Z disc. From Z disc to Z disc is your functional unit of the muscle fiber, right? And the Z disc to Z disc, this is called the sarcomere. From this Z disc point here, so let's say I go from this point here, from that Z disc, all the way over here to that Z disc. This whole length is consisting of the structural and functional unit of this myofibril, and that is called the sarcomere. Okay, that is our sarcomere. And the sarcomere is, again, the distance from Z disc to Z disc. Now, in that sarcomere, there's a couple other structures that I need to talk about. In this, we have this thick filament, this red thick filament, this big rectangular box. That big rectangular box there is called the thick filament. Okay, it's called the thick filament. So again, what is this red box right here? That right there is specifically called the thick filament. All right, now, the thick filament, if you look, here's this whole thick filament right here. This thick filament on this end, you see these blue, these nice little baby blue proteins right here? They're linking this thick filament to this Z disc. And the same way these proteins are linking this thick filament to that Z disc, the same situation is happening over here. Look at the end of this thick filament. You have these nice coily-like blue proteins linking the thick filament to the Z disc, as well as this protein is moving through the thick filament and linking it to these green proteins running right down the middle of the sarcomere. All right? These blue proteins right here are called titin. All right, titin. So this protein right here is specifically called titin. All right? It's called titin. So these blue proteins right here are called titin, and they're very special. We'll talk about why they're special in just a second, okay? But again, remember the titins are anchoring the thick filament to the Z disc. Also, they're connecting to these proteins that are running right down the middle part of the sarcomere. What are these proteins here called? Well, first off, before we name these proteins, what is this whole line here called? This line right here is specifically referred to as the M line. So these proteins here, all the way up, makes the M line. All right? So there's a bunch of different proteins, and as you look at, watch the M line as it goes up. M line goes up, goes up, goes up, and it connects to, what is this protein here? Titin. Okay, so if you look, the M line, which is running right up the middle of the actual sarcomere, which is consisting of two main proteins we'll talk about in a second, is linking the actual titin also to it. So if you look, this is connecting to the titin. What's the titin connecting? It's connecting the thick filament to the actual Z disc. So titin is helping to stabilize the thick filament, and it can do that by not only anchoring it to the Z disc, but also anchoring it to these accessory protein structure called the M line. What type of proteins is the M line made up of? The main one that it's made up of is called myosin. Myosin. So myosin is one of the main proteins that is a part of the M line. There's also C proteins. And there's another really important protein, which is called creatine kinase. But it's more of a functional protein rather than a structural protein. But they are scattered along the M line. So these are three really important proteins that are making up the M line. Again, one of the proteins is called myosin. The second one is called C proteins, and the other one is called creatine kinase. But again, creatine kinase, I'll star this one. And the reason why is because it's more of a functional enzyme. We'll talk about that when we talk about the metabolic actions of muscle fibers. Okay. Now, again, what's the function of the M line? The M line is responsible for connecting, these are accessory proteins, it's connecting the titin, and the titin is connecting the thick filament to the Z discs. So it's acting as accessory proteins that can help to stabilize the thick filament. That's their function. Titin, what did we say was the function of titin? It's actually stabilizing the thick filament by anchoring the thick filament to the Z disc, and it's also connecting with these accessory proteins of the M line. All right, sweet. Now, here's the next thing. From the end of this thick filament to the end of this thick filament, we call this a band. It appears like a nice dark band when you look at it. So what is this band distance right here? Let's say I come from this point about right down here. So I'm going to go from about right here all the way to about over here. This whole distance right here is consisting of what's called the A band. Okay, and the A band stands for anisotropic. Anisotropic. Here, I'll write that for you for those of you who want to know it. And all it means is it's just a darker. It means it's a dark band. Okay, so anisotropic just means it's a dark band. So the A band is the entire length of the thick filament. Okay, now from this thick filament end to this thick filament end over here, I'm going to come down here. So I'm going to go from this point here to about approximately this point right here. So now I'm going to come down from this point to this point. So I'm going to continue off of this point, and I'm going to come to about right here, approximately. Let's make this one blue. And we'll go to about right here. It's not perfect, but it's approximate. This is called the I band. Okay, this is called the I band. And the I band specifically stands for isotropic. Okay, so it stands for isotropic, which just means it's a lighter color. Okay, so it's the same thing over here. If we follow it over here, coming down from this point here, approximately, to about this point right here, approximately, this point right here is called the I band. And again, I stands for isotropic. If you want to know that, I'll write that down there for you. It stands for isotropic. And that just means, again, that it's a lighter color. Okay, so I'll write isotropic over here for you guys too. So far, what do we know? We know we have a sarcomere, which is from Z disc to Z disc. We know that we have an M line, which is a protein running right down the middle of the sarcomere, which is consisting of myosin, C proteins, and creatine kinase. We also know that you have this thick filament. And from one end of the thick filament to the other end of the thick filament is an A band, standing for anisotropic, which just means that it's a dark band. On this end of this thick filament to the end of this thick filament on another sarcomere. So for example, this is a sarcomere here, right? We'll call this sarcomere 1 or sarcomere 2. Over here could be sarcomere 1. So on sarcomere 1, from this end of this thick filament on sarcomere 2 to the actual end of this thick filament on sarcomere 1. That distance right there is the I band. And the I band stands for isotropic, which means it's a lighter color. Okay, now, if you look here, we have a whole bunch of proteins coming off of this bad boy right here. This big protein structure right here, we're going to call this whole thing the thin filament. Okay. So we're going to call this whole component here, this whole, all this blue, the orange, the green, the black, this is all the thin filament. All right. Now the thin filament is directly connected to the Z disc through this black protein that's running right down the actual middle of it. It's building the foundation for it. That black protein that's running right down the middle of this is called nebulin. So again, you can see it right here. So this is a thin filament. This is a piece of a thin filament. That's a thin filament, and this is a thin filament. And again, connecting this thin filament to the Z disc is this black protein right here. And again, what is that black protein there called? This black protein is specifically called nebulin. Okay, nebulin is a protein that's anchoring this whole thin filament structure to the Z disc. All right. Now, you see these blue proteins right here that's making like a nice little supramolecular helix? That's called actin. But here's the thing. With actin, there's two types of actin mainly that we're going to talk about. So let's say I take one of those individual structures there, just one individual circle. That one individual circle that kind of is like kidney bean shaped, if you actually look at it in a normal way, it's actually called G-actin. So a monomer of actin is called G-actin. Let me write that down. This is a monomer. But then let's say I polymerize this bad boy. And when it undergoes this polymerization, let's say I take a whole bunch of these bad boys, a whole bunch of these G-actin molecules and connect them together. If I start doing this now this is called F-actin. Okay, so now this is called F-actin. Now, what happens is when I take this G-actin and I make this new actin, it's called the filamentous actin, or I just like to call it the filamented actin, and then G stands for globular actin. So when I go from globular actin to filamented actin, it's just because I'm polymerizing these guys. So this is a polymerization reaction. Now all that happens is, to just be simple, let's say this is a monomer of F-actin, and I take another monomer of F-actin, and I take and make a helix-like structure. That's what you see here for this actin. Okay, so when you look at this actin here, it's looking like a supramolecular helix, which is consisting of multiple actin molecules. Okay, so this blue structure right there is specifically called actin. All right, so that's our actin molecule. All right, so we got actin here. And again, nebulin is this black protein anchoring this thin filament to the Z disc. Actin is this whole little supramolecular helix consisting of, again, G-actin being polymerized into F-actin, and then multiple F-actins linking together to make this supramolecular helix. Then, you see this orange protein right there? This orange protein is like a wire. So here, I want you guys to imagine something for just a second. Let's say here, in each one of these F-actins, I have a little hole, and that hole is where this protein called myosin binds. Well, there's that orange protein, and what that orange protein does is it actually wraps around these holes and blocks the myosin head from being able to bind. So what this protein is doing is it's coming and surrounding and blocking these holes. What are those holes called? These holes are called the active sites. So what this rope-like protein is doing is it's coiling around this actin and blocking the active sites in a resting position. So whenever the muscles are resting, this protein, this rope-like protein, is blocking the active sites. What is that rope-like orange protein called? It is called tropomyosin. So look at this orange protein right here. This is called tropomyosin. Okay, so tropomyosin. And again, what is the function of tropomyosin? Tropomyosin, normally when the muscle is at rest, is blocking the active sites on actin, preventing this myosin head that we'll talk about in a second from actually binding. Okay, so now, if we have this tropomyosin, again, what is it doing? Just one more time. It's acting like this red rope protein, right? And again, this is actin. These little holes are the active sites. That's where the myosin head that we'll talk about in a second binds to cause contraction. When our muscles are at rest, this cord-like protein called tropomyosin is blocking the active sites, preventing the myosin from being able to bind on. We'll talk about what allows for them to move and open up those active sites for the actual myosin heads to plug in the neuromuscular junction video. Okay, so that's our tropomyosin, which is a part of the actual thin filament. Now if you look here, we have this little green protein. Let me expand a little bit on this green protein for a second. Okay, let's say here I have this green protein, and there's 3 different sites on this green protein. One of the sites is binding to this blue protein that we called actin. The other site is binding to the orange protein that we called tropomyosin. So if one over here is binding to, again, what would this one be binding to? It could be binding to actin, or the other one could be binding to tropomyosin. And then this part here is specifically binding to calcium. Okay, so this has 3 different sites. In other words, we're going to call this whole molecule, this whole green protein molecule, we're going to call it troponin. Okay, this is called troponin. This whole big molecule up here is called troponin. But now we got to be a little bit more specific. The site on troponin where calcium is binding is called troponin C. That's easy because there's calcium, and calcium starts with a C. So this point here is called troponin C, but I'm going to put just T and C. Okay? So that site right there where the actual calcium is binding is called troponin C. This point right here, where the actual troponin is binding to the tropomyosin, is called troponin T. And that's simple because tropomyosin starts with a T. So this site right here is called troponin T. Now here's where you guys would be like, oh yeah, this one must be troponin A. That's wrong, all right? It's not troponin A. It's actually called troponin I. And the reason why it's troponin I is because I stands for inhibitory, okay? And we'll talk about that more in neuromuscular junction. I just want you guys to get the structure down first, because if we get the structure down pat, we're really going to understand the function. Okay, so again, this whole green protein molecule is called troponin, but there's 3 different sites on this troponin. One that binds to the tropomyosin is called troponin T. The part that binds to the calcium is called troponin C, and the part that's binding to actin is called troponin I. Okay? And these are good markers for certain types of heart damage, right? So if someone is having a heart attack, some of these actual proteins might leak into the actual bloodstream and could be an indicator for a myocardial infarction, which is a heart attack, right? Specifically in cardiac muscle, but we're talking more about skeletal muscle. Okay, so that covers that part. So again, what is this green protein here called? That is called troponin. All right, so we've pretty much covered the thin filament. Let's review the thin filament one more time. Black protein is nebulin. This blue protein is actin. The orange protein is tropomyosin, and the green protein is troponin. Okay, we covered the thick filament a little bit, but now we have to go into a little bit more detail on this thick filament. This thick filament is having coming off of it this little like tail. So you see this little like tail right here? It looks like it's coiling. That's because it is. There's 2 protein chains. So this is actually going to have this little protein chains, this little alpha helix-like structure. So there's like a little alpha helix here. Okay? And what happens is, this is the tail part of myosin. So this whole protein here is called myosin. So again, what is this protein here called, guys? This whole protein right here is called myosin. Now myosin is made up of 3 parts. This part right here is called the tail. Okay, it's called the tail. This mid part here is called the neck. And this top part here, it looks like a double like head. Like for example, if I were to kind of show it like this. Let's say I have here's that part, the tail. here's the neck, and there's two heads, okay? So again, this part here is the tail. This part here is the neck. And this part here is the head. Why is that important? The head is what binds into these actin active sites. So you see these little holes right here in the actin? That's what the head binds into and allows for this moving or sliding of these myofilaments. Okay, all these myofilaments are just all these protein structures. Okay? That's the function of the head. Another function of the head is that it has an enzyme component on it. And we'll talk about that more, but it's called a myosin ATPase. In other words, it can cut ATP and break it into ADP and an inorganic phosphate. So it has what's called ATPase activity. It can cleave ATP into ADP and inorganic phosphate, which is also important for the sliding of the myofilaments. The neck region is important. And the reason why the neck region is important is that there's what's called light chains. So let's say here I have a blue chain here. We call that a light chain. And then there's another one. Let's say I have another chain over here binding to the other side for a second. These light chains are really, really important because these light chains, not only do they support the actual head and the neck region of this myosin, but they also can undergo regulation. So for example, when phosphate ions bind onto these light chains, for example through myosin light chain kinases, that can also control the activity of this myosin molecule. So again, myosin is consisting of a tail, which is looking like an alpha helix, has a neck, which consists of light chains on it. If you really want to know the light chains, you have what's called regulatory light chain. Okay, that's one of the actual light chains. Let's just say that's the green one. The other one is going to be the essential light chain. Okay, so the regulatory light chain is what can undergo phosphorylation and help to change the activity of the myosin. The essential light chain is more structurally stabilizing the myosin head and the myosin neck. Okay, so we got the myosin now. Okay, so in short here, what do we have so far? What have we been able to cover? We have the Z disc. I didn't tell you what the protein that's making up the Z disc. There's many proteins, but the main one that's making up the Z disc, the main protein, is called alpha actinin. Do not get that confused with actin. So the main protein of the Z disc is the alpha actinin. That is the main protein of the Z disc. All right, so now let's do a quick review of this real quick, all right? This whole big myofibril structure. So one more time, we have the A band, which stands for anisotropic. What does anisotropic mean? It means that it's darker. What is it primarily consisting of? The thick filament. All right, so it's going from this end of that thick filament to this end of that thick filament on the same sarcomere. What determines a sarcomere? It's from Z disc to Z disc. That is our sarcomere, okay? I band stands for isotropic, which means it's lighter colored. What is that consisting of? If we come up here, it's going from the end of this thick filament on this sarcomere to the end of this thick filament on another sarcomere adjacent to it. That is the actual I band. And it's primarily consisting of what? If you think about it, if it's only taking this distance, it's only consisting of like the Z disc, it's only really consisting of this thin filament proteins, right? That's all it's really consisting of. So really, if you think about it, the I band is really only consisting of the thin filaments, and it's also consisting of like the titin and a little bit of the Z disc structure, right? Then we have the M line. And the M line is these actual made up of myosin proteins and C proteins and creatine kinase enzymes that are running all the way up through the middle of the sarcomere, okay? And again, what are they doing? They're linking to this titin structure. What is titin doing? Titin is anchoring the thick filament to the Z disc. On top of that, titin is also connecting to this M line proteins to stabilize the structure of the thick filament. Okay. Oh, we got another one that we have to cover. The distance, I'm going to get rid of this part here. The distance from this thin filament to this thin filament over here. What is this right here called? So the distance between this thin filament and this thin filament on the same sarcomere is called the H zone. This is the H zone, okay? So again, one more time. H zone is the distance from this thin filament to this thin filament on the same sarcomere, okay? All right, so we got that. Then what else do we say? We have the thin filament. What are the main proteins of the thin filament? The blue protein is called actin, okay? And what do we say about actin? Actin is originally in monomeric form called G-actin, which stands for globular actin. When you polymerize a bunch of those G-actins, you get F-actin, which just stands for filamented actin, right? This filamented actin can actually it can act as a monomer, bind with another monomer, and form this supramolecular helix. And this reaction occurs by polymerization. The other protein was this orange protein, which is called the tropomyosin. Tropomyosin is responsible for covering the active sites of the actin during rest, so that the myosin heads can't go in there and bind in. But whenever we have a situation, what is that other protein that we talked about? We talked about troponin. Troponin has 3 different sites that you can bind to. One is when it's binding to tropomyosin. The other site is when it actually can bind to calcium. And the other one can bind to actin. If it's binding to calcium, it's called the troponin C site. If it's binding to tropomyosin, it's the troponin T site. If it's binding to actin, it's called the troponin I site. A real quick prelude into what we'll talk about in the excitation-contraction coupling is when calcium binds onto the troponin, right, it changes the shape of the troponin T component. And when it changes the shape of the troponin T component, it pulls on the tropomyosin. And when it pulls on the tropomyosin, it opens up those active sites for the myosin head to plug in, okay, and then cause the movement of the myofilaments. Then we said that this black protein that's running from the Z disc all the way through the length of this thin filament is called nebulin, and it's acting as a supportive structural protein. Then we said that this thick filament is having a protein coming off of it called myosin. And again, myosin has a tail region, it has a neck region, and it has the head region. Head is responsible for binding into the active sites of the actin and moving the myofilaments. It also has an ATPase component that it can cleave ATP into ADP and inorganic phosphate. And the neck can have proteins bind onto it that around this, which are called light chains. And there's 2 different types of light chains. Regulatory light chain, which can bind the phosphate and change the overall shape to play a role in contraction, or essential light chain, and that stabilizes the neck and the head. All right? And then again, we said that we had the H zone, which is the distance from this thin filament to this thin filament. Now I need to talk about one last filament, which is so extremely important. We need to talk about this one. Okay, so let's say over here, I have this actin. I have another protein, a very, very important protein, that connects the actin. Let's say here's this protein right here. And what this protein does is it links the actin to the extracellular membrane. So what is that? Let's say here we have the membrane of this muscle cell. Here's the membrane. Okay, and you know the membrane is actually consisting of a phospholipid bilayer. So let's say here is the actual muscle cell membrane. There's so many different proteins that this dystrophin is linking to on this sarcolemma. So what is this structure here called? It's called the sarcolemma. Okay? There's so many different proteins. I'm just going to draw a crude diagram right here. And I just want you guys to understand that there's this whole protein complex here. And what's happening is dystrophin is linking the actin. You see here's this blue protein called actin. Dystrophin is linking actin to the actual cell membrane, to this whole protein complex. And what happens is outside of our cell, we have an extracellular matrix, which is consisting of a lot of different types of fibers. What happens is these proteins can link to this extracellular matrix. So now imagine what would happen, because I hear this a lot, at tutoring, is that people will constantly say, all these proteins, what do I really need to know them for? They're not even relevant. They are. And the reason why is look at this one protein, this one little protein that we're talked about called dystrophin. So again, what is this protein here called, this green protein? It's called dystrophin. And what dystrophin does is it links the actin to the sarcolemma through these actual protein complexes, which link those proteins to the extracellular matrix. If there is a situation in which there is a mutation in the dystrophin gene, and they don't produce any dystrophin, this can lead to what's called a muscular dystrophy. So now what I want to talk about is I just want to do a little tidbit on situations in which this dystrophin protein is actually either not present or it's mutated, because it's all clinically relevant. Because being a good, either doctor or PA or nurse, whatever you might be going into the medical field, it's not just good enough to know the physiology. It's really important to know what happens if this protein breaks down or if this protein doesn't work. That's where it's really clinically relevant. So I'm going to just provide a little tidbit. In the situation in which this actual dystrophin protein isn't produced or it's mutated, it falls under an umbrella of what's called muscular dystrophy, right? So there's many different types of muscular dystrophies. We're going to talk about the two most common ones. So out of the many types, we're going to talk about Duchenne really briefly, and then we're going to talk about Becker. Now, out of these, to keep it super simple, muscular dystrophies is usually an X-linked. That's an X-linked recessive disorder. So it's usually an X-linked recessive disorder. In other words, it's more common in males. So males are more likely to develop muscular dystrophy as compared to females. And the reason why is males only have one X chromosome. Females have two X chromosomes. So if they do what's called, they inactivate lionization of the actual other X chromosome that they have, there's less likely of a chance that they'll develop this muscular dystrophy. Now, Duchenne muscular dystrophy is much, much more severe. Okay, in this situation, the individuals who develop this type of muscular dystrophy is usually they develop it by the age of like 5 or 6 years old. Okay, so usually these individuals develop it by the age of like 5 or 6 years old. Now, what is the difference then? If really it's an X-linked recessive disorder and it's due to the dystrophin gene, what's really the difference between Duchenne and Becker? Well, Becker is not as severe, it's less severe. It's still bad. It's still a terrible disease. It's less severe, but it takes a little bit longer for the actual onset of the disease to develop. So usually this develops by the age of like 10, and we give it a nice range to about 20 years old. And here's the real big difference genetically. I don't want to get into the depths of genetics. We'll talk about this more in detail in a pathophys video. But if you don't produce the dystrophin protein, so just no dystrophin protein, that's Duchenne's, okay? So the lack of dystrophin, so no dystrophin produced, causes Duchenne's muscular dystrophy, okay? So usually this is a result of what's called a nonsense mutation. A nonsense mutation is when you have a premature stop codon in the mRNA and the protein doesn't completely develop. The second type is a frame shift mutation, where there's like an insertion of an extra nucleotide or the deletion of a nucleotide. And that can result in changing the reading frame and result to the inadequate production of a functional protein. So this is causing Duchenne's muscular dystrophy. Becker is usually due to what's called a missense mutation, where you're just substituting an amino acid in for another one. And it changes the overall shape. And if you know, there's what's called the complementarity concept, that structure complements function. So whenever the protein shape is changed from its normal structure to an abnormal structure, it changes the function. So in Becker's, it's due to a misfolded protein. So we say a misfolded protein because it's caused by a missense mutation. So this is caused by a misfolded protein. Now you guys are probably wondering, okay, well you told me all about muscular dystrophy. How does it actually happen? Well, imagine here. This is anchoring this whole structure to this actual cell membrane. If this protein isn't present or it isn't functional, what's going to happen to this now? Now this membrane is going to sag down. If this membrane starts sagging down because it doesn't have this dystrophin to hold it stabilized and steady and maintain the cytoskeletal structure, again, what will happen? This membrane will sag down. Over time, the membrane will start to break down. As the membrane starts to break down, proteins and enzymes and ions start moving in and out of this actual cell. And eventually, the muscle cell becomes fibrous and fatty. So in other words, the muscle cells die, they get replaced with fibrous tissue and fatty tissue, and they don't function. So what would eventually happen to people with muscular dystrophy? They have muscle weakness. And this muscle weakness can get so bad because one of the main skeletal muscles is called our diaphragm. The diaphragm is responsible for helping us to breathe and inhale. So if this muscle was actually damaged or became progressively weak, they would develop respiratory failure. Another thing, they'd have a hard time being able to move. They usually have a strange waddling gait, right? And it's because they don't have very, very strong hips, right? So usually the muscles of the hips are very weak. Another thing that can also be a problem with this muscular dystrophy is eventually if it can affect the heart muscle, because dystrophin is not only present in the skeletal muscle, but it's also present in the heart muscle. So it can cause the muscles, the actual heart muscle, to become very, very dilated and very floppy and flabby, and that's called dilated cardiomyopathy. So usually these individuals, there's not much treatment for these individuals. They have to get physical therapy to try to improve the quality of life. Sometimes they can get glucocorticoid therapy, but there's a lot of side effects. And sometimes they're actually working on stem cell therapy also, but not much success so far in that, unfortunately. But it just goes to show you, these proteins, they play such a huge role. Something so small can play such a big, big role in our entire body's physiology. Okay, so that pretty much, guys, covers a lot of information here about our actual myofibrils, consisting of our sarcomere, our A bands, our I bands. We'll talk about this again. We'll actually recap this even more whenever we go into the actual excitation-contraction coupling. Guys, I really hope you guys enjoyed this video. I hope it made sense. Thanks for sticking in there with me. I know it was a long video. I hope you guys really did enjoy it. If you did, please hit the like button, subscribe, put some comments down in the comment section. All right, Ninja Nerds, until next time.